So today we're going to discuss about glycolysis process, and as well as we'll get to know about gluconeogenesis, as well as the topic about glucogenolysis. So as the name says, glycolysis means breaking down of glucose into two pyruvates in fed stages, which is known as the stage when we eat. So that's the stage we call it as a fed stage. So these stages will happen in the liver. So when we eat food, the glucose will be there, and the glucose will be traveled towards the liver with the help of blood, and in liver there is a catcher which will catch glucose, and it's known as blood type 2 transporter, which is responsible for bringing the glucose inside the liver, and it's also responsible for letting the glucose out whenever it is necessary from liver. So once the glucose will be catched with the blood type 2 transporter, uh, we have an enzyme known as glucokinase, which is responsible for adding phosphate in the carbon position of glucose. In glucose, we have 6 carbons. So at the sixth position, the glucokinase will add the phosphate. And we also have another enzyme which is known as glucose 6 phosphatase, which is responsible for removing this phosphate carbon, this phosphate in the sixth position of the glucose. So after adding this after adding the phosphate to the glucose, then liver decides to convert this glucose as fructose. Same structure, just the name is changed over here. And this fructose is also known as, generally we call it as a fruit sugar. So after the glucose 6 phosphate converts into fructose 6 phosphate, liver again decides to add one more phosphate in the first position of fructose, and the name will become as fructose 1,6 base phosphate. BI means two phosphates which is present in the fructose structure, and the S will indicate they are separate. So by this we'll understand there is a one phosphate which is positioned at the first carbon position of fructose and at the sixth carbon position of fructose. So and again fructose 1,6 bis phosphate will again converted into two byproducts which are known as glyceraldehyde 3 phosphate and also dihydroxyacetone phosphate. Their structure will be same, but they are splitted. They are slightly splitted into two halves, which has two carbon atoms and one phosphate within them. And again, this glyceraldehyde and dihydroxyacetone will convert as 1,3 bis phosphoglycerate, and here also it will again the second one, which is dihydroxyacetone, it will also convert as 1,3 bis phosphoglycerate, and again due to some reasons these has to turn into pyruvate. Until the pyruvate, this cycle will repeat. So at first it became the 1,3 bis phosphoglycerate, and again it will become 3 phosphoglycerate, which has 3 phosphate atoms, and afterwards it will become as phosphoenol pyruvate, just a product just before pyruvate. Okay. And again, the phosphoenol pyruvate will convert as the pyruvate, and this pyruvate can be, go through the Krebs cycle, and ATP will be formed. So this is all about this glycolysis. Now we'll understand what happens at gluconeogenesis. Gluconeogenesis is a process to make glucose atom when glucose is not present. When there will be no glucose? Fasting. During the fasting. Fasting which is more than 6 hours. Here the non-carb structures will be broken down and they will convert as glucose. Non-carbon structures such as amino acids, fatty acids, these are all are known as non-carbon structures, carb structures, sorry, not carbon, carb structures, and they will become glucose. So, there are glycogen, which is a polymer of glucose. This will be stored in two areas. One is in the liver and another is in the muscle. In liver, it will be stored in heptic cell or also known as liver cell in a polymer position, and heptic cell has a capacity of storing glycogen. 80 to 100 grams of glycogen can be stored in heptic cell or in the liver cell. And in muscle, 250 grams to 400 grams of glycogen can be stored. So let's understand about how the glycogen can be removed. At first, the glycogen will be removed from the liver. Then, after when there is no more glycogen left in the liver, the muscle will be forced to release this glycogen to form glucose. So once everything will be done from the liver, like the muscle will convert glycogen into glucose 6 phosphate, and since there is no more enzyme which can remove this sixth photon phosphate, which can remove the sixth phosphate, it has to send this thing to liver. So when the glycogen will convert into pyruvate in the muscle, there will be another two more byproducts will be evolved in this process, which are lactate and alanine. And the lactate and alanine pyruvate alone, they will flow in the bloodstream and they will reach the liver. And in liver, we have an enzyme which will recognize lactate. It's known as lactate dehydrogenase. And this enzyme will recognize the lactate and forces lactate to convert into pyruvate or the product which can be make glucose. And along with alanine, these will be converted, and the glucose 6 phosphate will be formed, and they will become glucose, and the body will get the needed glucose. And this will happen when you're doing heavy intensive workouts for 27 minutes. So after 6 hours of fasting, we need 27 minutes of high intensity workout, so we can burn or we can oxidize all the glucose then which are present in your liver and in the muscle, so that the glucose will be developed and clients will lose glucose, the excess glucose whatever stored in our body. And here comes the facts. In our body, the fatty acids are stored as triglycerol, and we have a glycerol bone. At first, the glycerol bone will be separated from the three fatty acids, and the glycerol bone will be forced to go inside the liver. And there is another enzyme which is known as glycerol kinase. And this glycerol kinase will recognize the glycerol bone, and it will convert him into, and it will welcome him. Let's say it's as a welcoming. It will welcome into the glyceraldehyde family, and the process will start from here, and then it will be the process will be reversed. The glycerol dehyde will come to fructose, and the fructose 1,6 bis phosphate, and it will be turned into fructose 6 phosphate, and it will turn into glucose 6 phosphate. And here we have a glucose 6 phosphatase, the enzyme which will remove phosphate gland, and the glucose will be formed. And here our gatekeeper, who we call him as a blood type 2 transporter, or whatever you call him, GLU type 2 transporter, and he will let this glucose into the bloodstream, and the glucose will reach the 5 organs which are highly dependent on glucose, which they need 24 by 7. You can call it who is known as central nervous system, retina, testis, kidney. So these are the glands and RBC. So they need the glucose. So that's the reason why this process will happen.